Okay, so today's lesson is about fungi, and fungi we've talked about before because we talked about using the single cellular fungus, the yeast, as part of biotechnology. Remember that fungi are mostly multicellular except for the yeast, and that their job in ecosystems is to be decomposers. What I want to do is focus you in now on the cells of fungi and how they're different from the plant and animal cells that we've been talking about. Fungi are eukaryotic, so the only prokaryotic cells we deal with are the bacteria, and we've kind of already dealt with those. So let's dive into fungus, and before we dive into their cells, we have to talk about how the fungus actually operates. And what you see in front of you is a picture of a fungus. This could have been a mushroom, it could have been any number of fungi. This one, instead of having the cap kind of turned up, the cap instead of the cap being pointed up, or down, the cap is pointed up. What you need to understand is the part that we normally think of as the fungus is this, right? That part is called the sporangium. Running out of space here, but sporangium. In other words, it's the spore delivery system. It's the reproductive part of the fungus, and actually only the reproductive part of the fungus. The body of the fungus is under the surface of the ground, or if you're talking about something like a mold, it's under the surface of the bread or the orange or whatever's molding. What is the body of the fungus is called mycelium, and that's what this is. Alright, so this is the body of the fungus, the mycelium. So the fungus actually lives underground. The mycelium is a network of thread-like extensions called hyphae. So each individual little thread here, each one of these is called a hyphae. Now what happens is the hyphae penetrate a food source. So remember, they're decomposers, so they kind of penetrate something that's dying or dead and decomposing, like say an old tree branch. So they penetrate that tree branch, and the hyphae excrete digestive juices from the tip of each hypha. Okay, so right here at the tip, they start to excrete digestive enzymes, and those digestive enzymes start to break down the dead, rotting wood, and then the fungus absorbs all the nutrients that are broken down from that. And it's very important that I tell you that, because this is going to have a bearing on how the cells of a fungus operate. The other thing that's important here about the fungus is that the growth only occurs at the tip of the fungus. So the cells back here in this part of the hyphae back here, these cells are done growing. Growth occurs only at each little individual tip that you see here. Each of these tips are where the fungus is still growing, so it spreads out from the tip. Alright, so now let's talk about the cells of the fungus, because that's where we're headed. So we're going to take this hyphae here that you see, and we're isolating just the tip of one of the hyphae. And what you can see are several cells of the fungus here. So this is cell 1 right here, and this is the cell at the tip, cell 2. One of the things that you'll notice as you look at this picture, one major difference between the cells of a fungus and the cells of a plant or an animal are that look at what this plasma membrane does. Normally, plant cells and animals have a membrane that surrounds each cell, right? So if you have, say, a plant cell like this, the membrane of the cell would be all inside, surrounding each individual cell, and then an adjacent cell would be here like this, right? And the membrane would come around and surround each individual cell. So if this were a plant root, each of these would be stacked together like that. What you'll see here is, notice that there's one continuous plasma membrane that goes around the entire hyphae. So all of the cells share one plasma membrane, and there's no break in the plasma membrane here between cells. Also notice there is a cell wall on the outside. So this is surrounded by a cell wall, and you'll have a thick part here between cells, right here and here. That's called the septa. So that's another difference. So you have one cell wall that surrounds everything as well, and divisions between the cells occur because there are septa. And you'll notice there's a space in the septum. and that's a little pore so that the cells can send their cytoplasm back and forth. So if this cell at the end absorbs something, it can pass the nutrients on through to all the other cells all the way down the line. So that's another big difference. So we've got the plasma membrane goes all around all the cells. We've got the septa that divide the cells, so there's no cell wall that goes between them. It only goes around the outside. And of course, because these are fungi, people often make the mistake of thinking that they're going to do photosynthesis. Remember, fungi are decomposers. They get their nutrition by decomposing dead things. They have no chlorophyll, no chloroplast. They don't do photosynthesis. These are consumers, okay? Decomposers that are consumers. They're not like plants at all. Even though these hyphae resemble plant roots, they're not the same, okay? The other thing you'll notice is that in a fungus cell, you'll have several nuclei. If you look at this one on the tip here, there's a nucleus here, here's a nucleus here. That's called being multinucleate. So every cell has a couple of nuclei in it. So that's another major difference that you're going to see. The last thing is that here at the tip of the hyphae, you'll see these little secretory vesicles. Remember I said the tip penetrates a food source and secretes enzymes that break down the food source. So these little vesicles here in the tip will actually kind of be pushed to the end and open up into the food source so that all the enzymes go out into the food source. Now remember, these cells communicate with one another. There's no space between them. So what can happen is you can make, way back here, you can make enzymes and put them in little vesicles, and those vesicles can actually travel through the cells and end up here at the tip, and so you'll end up with a bunch of these secretory vesicles at the tip. So those are our main differences. Just to recap then, we've got cell wall that goes around all of them, right? Inside of that cell wall, you've got a plasma membrane that surrounds all of the cells with no divisions in between. You've got the only separation between the cells being the septa, and those septa don't go completely across. There's a hole in the septum to allow movement of cytoplasm. Secretory vesicles might be made in other cells, but then they get sent to the very tip. And they're multinucleate cells, so you have multiple nuclei. The last thing I forgot to mention as I'm looking at my list here is the makeup of the cell wall. Like plant cells, they have a cell wall that we've talked about, but the cell wall here is made of something called chitin. Okay, so instead of it being made of cellulose, the cell wall of a fungus is made of a substance called chitin. It's another complex carbohydrate, and that's spelled with a CH. C-H-I-T-I-N. Chitin. Okay, so that's the major difference. We've looked at all of the eukaryotes now, and we've compared them to plants and animals.